In this video, I will be talking about the concepts of biosynthesis of purine nucleotide. So before that, I think we have to understand the basic concepts. So what is purine? You know that purine is, it can be adenine and it can be guanine. But now we are synthesizing purine nucleotide. What do you mean by purine nucleotide? Means you have got a purine, that means you got a base, you got a phosphate, and you have got one of the purine. It can be adenine and guanine. Suppose if it is adenine, it is adenosine monophosphate. If it is guanine, guanosine monophosphate. And now our aim is to synthesize this adenine monophosphate and guanosine monophosphate, right? Guanine monophosphate. So now let us understand the concepts here. So the first thing written in our notes is biosynthesis of purine nucleotide. The first is de novo synthesis. Before entering the concepts of this, without knowing the word de novo synthesis, it's waste of understanding. So first, let us understand the meaning of de novo synthesis. What is de novo synthesis? De novo synthesis means it refers to synthesis of complex compound from simple molecules such as sugar or amino acids. That means we got this process of de novo synthesis. What is it telling that? It refers to synthesis of complex molecules. In our case, the complex molecules adenine monophosphate and guanine monophosphate. And we have to prepare it from either from the sugars or the amino. But so let's go with the first action of the de novo synthesis, right? So you have to understand regarding this, and the few points written in our notes is de novo synthesis main and difficult and regulated pathway. This is the word. You can see the main and regulated pathway here, and you can see multiple steps are involved at this aspect, right? And you have to remember that you have got something known as amphibolic enzymes that we'll be discussing, but right now let's understand the concepts here. As I told you, de novo synthesis is the formation of complex molecule from the sugars. From now let's take a sugar molecule, and that sugar molecule is ribose 5-phosphate. From where did you get this ribose 5-phosphate? The ribose 5-phosphate you got it from the HMP shunt, right? HMP, you understood that in the glucose metabolism we spoke about this HMP pathway. In that process we have taken from that HMP, and right now we have got ribose 5-phosphate. And thing you have to understand here is this ribose 5-phosphate converts into phosphoribosyl 1-pyrophosphate. The enzyme involved for the conversion of this ribose 5-phosphate to phosphoribosyl 1-pyrophosphate is phosphoribosyl pyrophosphate synthase. It's very simple. It's synthesizing this, so we have called this as synthase. Another thing you have to understand, if you see the difference here, 5-phosphoribosyl pyrophosphate and this 5-phosphoribosyl 1-amine. If you understand, if you read carefully here, this phosphate is replaced by amine. Right now, who is donating amine? It is glutamine, which is donating amine, and it has become glutamate. In the presence of an enzyme, glutamine phosphoribosyl amine transferase. You can observe this. This glutamine is transferring the amine, so we call it as glutamine phosphoribosyl amino transfer, because it is transferring the amine group to this phosphoribosyl pyrophosphate. So that is the reason we have named it as glutamine phosphoribosyl amine transferase. So now let us understand that this 5-phosphoribosyl 1-amine, it undergoes multiple steps. Right now you understand that IMP monophosphate, it undergoes two equations. One in the presence of dehydrogenase and others in the presence of adenylyl succinate synthase. So what happens in the presence of IMP dehydrogenase? It forms XMP, xanthine monophosphate. We'll be dealing that. The xanthine monophosphate converts into guanosine monophosphate, and this is what we need, and we got it. And what is the other reaction? The other reaction is adenylyl succinate synthase, and it forms adenylyl succinate. It converts into AMP, and this is what we need. So the whole concepts of de novo synthesis is formation of these complex substances like GMP, AMP from the sugar molecules or amino acids. But in this case, we are taking the sugar molecule, where you can observe carefully this ribose 5-phosphate is converted into 5-phosphoribosyl 1-pyrophosphate. Let's not by heart here. Next, the glutamine came and it has donated amine to this, and now we have got 5-phosphoribosyl 1-amine. and now it converts into inosine monophosphate, and this inosine monophosphate converts into, in the presence of enzyme dehydrogenase, converts into xanthine monophosphate and guanine monophosphate. And this adenoate succinate, with the presence of this enzyme adenoate succinate, it converts IMP to IMP to AMP. This is all the basic concept you have to understand, and that's all it's written in our notes. And one thing you have to understand, the enzyme, right? This enzyme glutamyl amine transferase, this PRPP, right? And this enzyme is rate limiting enzyme. Rate limiting enzyme. This is what you have to understand. Why there is rate limiting? You can see all these GMP, GDP, GTP all are negatively inhibited by this. And you can see even this AMP, ADP, ATP, all which are formed from the respective compounds, they inhibit, they have got a negative action on this enzyme. So this is a rate limiting enzyme here. This is what you have to understand, right? And that's all it's written in our notes. So now let us understand the basic concept of this. What is salvage pathway? What is salvage pathway? Before that, let's understand the concepts of salvage pathway. Salvage pathway is a pathway in which nucleotide, purine or pyrimidine are synthesized from intermediate in the degraded pathway for the nucleotides. Salvage pathway is used to recover base and nucleoside that are formed during degradation. So what is it trying to tell us? Right? What is it trying to tell us? So let's know the equation of these salvage. Let us know them and let us understand cleanly, right? In the sense with concepts. Right here. So the first thing you have to observe, one thing I want to tell you. See, you can see this is guanine, right? You can see the structure of guanine. You have gotten, right, 2 rings. You can see the structure of this. What is the difference between guanine and this? Well, you have one ribose sugar and you have got one phosphate. So we need some enzyme which helps the guanine, or which transfer this group, ribosyl and phosphate group, to the glutamine. So that's all you have to understand now. So now let us understand the concepts of salvage pathway. If you see the concepts of salvage pathway, the first one is adenosine is converted into adenosine monophosphate. So if I show you the structure of adenosine, this is adenine, right? You can see the structure of adenine here, adenine, right? If you see the structure of adenine here, purine, it is converting into AMP, adenosine monophosphate. Why? Because adenine with the sugar, it is known as adenosine and it has got phosphate. Who is helping that? It is this adenine pyrophosphate, it is adenine phosphoribosyl transferase. So what do you mean by phosphoribosyl? It means it is this enzyme is transferring phosphate and the ribosyl to the adenine. So that is the reason it is known as adenine phosphoribosyl transferase. So now you understood the concepts. Yes, this adenine, when who is donating that ribophosphate, it is this PRPP. It is PRPP. What is PRPP? PRPP is nothing that it is phosphoribosyl 1-pyrophosphate that is donating these phosphate and 1 ribose to the adenine, and you have got AMP. If you see the next equation, that is so let's see the guanine first. The guanine is converted into GMP. If you see the structure of guanine, this is the structure of guanine, guys. And now if you see the structure of GMP, the difference between the guanine and this is it has got ribose and phosphate. And who is transferring this phosphate and sugar molecule to the guanine? It is none other than same, you have got phosphoribosyl. Since it is transferring phosphoribosyl, it is known as phosphoribosyl. It is transferring to xanthine, so it is known as xanthine phosphoribosyl transferase. This enzyme also used in the conversion of guanine to guanosine monophosphate. How does it convert? This enzyme takes one phosphate and ribose from this PRPP and it donates to guanine. So it converts into guanosine monophosphate. And the same thing is that this enzyme it takes one phosphate and ribose from this PRPP and it donates to xanthine. And now you have got a phosphate and the ribose in the hypoxanthine. It is known as IMP now. It is known as IMP plus phosphate. So this is all you have to understand regarding this salvage pathway. the basic concept here is you have to see, if you observe carefully here, the same enzyme is involved, phosphoribosyl, and this is also phosphoribosyl transferase. You can see this, here is an adenine, that is the reason you wrote adenine. It is a hypoxanthine, that is the reason you wrote hypoxanthine. If you see guanine, that is the reason you wrote guanine. So these all you have to understand first, right? Right. So now let's end this. We have to speak about this also. So this is the minor pathway. Available rings is used here. What is the source? From the diet, primary source like cereals and pulses. Secondary source, it can be the source of the adenine and guanine. It can be from DNA, RNA degradation. And these are the reactions we already spoke. By important things you have to understand. This is very important enzyme. This is hypoxanthine guanine phosphoribosyl transferase. Right. So this is all you have to understand regarding this de novo synthesis and the salvage pathway. The other thing you have to understand here is, if you see the structure, right, the structure of purine is important. They ask you questions here. The questions they ask you here is, which atom is donated by whom? Suppose if you see the structure of this purine here, so let me make clear here. So if you see the structure of purine here, this is 1 2 3 4 5 6, you can see 7th atom, 8 9. So now let us understand who is donating what. If you see the first one, the nitrogen is donated by aspartate. How do you remember? 1 in alphabetical A comes in first, so aspartate. And one thing you have to understand, before to understand this, you should always know that 1 3 9 7, this location we have nitrogen. So you have got 1 3 9 7, right? The other thing you have to understand is, before going in detail, right, you know that 1 3 9 7, just write the nitrogen first. Then you can see this, just take this 3 away, and these all are donated by this glycine. All these are glycine. So what is left? We have finished aspartate, we have finished glycine. And now these two nitrogen which are below, right, they are donated by glutamine. Just remember glutamine, you're sticking it down, so glutamine. Now let's see about, we already spoke about this. Now let's see about the carbon which is donated to the second and the 8th. The carbon donated is N10 formal THF, and this is N10 formal THF, right? So this is what you can understand. This is what you have to remember. They ask you the contributions also. So this is what for now. Let's meet in the next session. Thank you.